We have heard of many interactions where microbes, they are interacting with plants, where they are interacting with animals, and they are doing multiple of things for them. Sometimes we know that they are sitting in our intestines and they are synthesizing vitamins for us. We know that they are like colonizing the roots of the plants, and they are absorbing the what you call as nutrients and water for these particular plants. They are fixing the atmosphere in nitrogen, they are degrading the waste, they are producing so many things. Matlab, this is what something we casually know about the microorganisms, right? One more interesting thing matlab, these microbes do, not one more, there are many more things which these do, but then today what we will be talking about is only about one particular thing, about one particular species of a marine bacterium, a gram-negative rod-shaped marine bacterium, well known for its phenomenon to emit light, the phenomenon which we call as bioluminescence. Matlab, the Allovibrio fishery, earlier this was classified as Vibrio, but later on, like when molecular methods came in, this was reclassified as Allovibrio. So this Allovibrio fishery is very famous for being a, you can say, a mutualistic symbiotic associating bacterium with the Hawaiian bobtail squid. So here what it does, it is not providing nutrition to the squid, but then yes, it is protecting the squids from its predators. It does not kill its predators anyways. What this particular bacteria, the Allovibrio fishery, is going to actually do is, this particular bacteria is going to provide the counter illuminating camouflage. Now, as we know that this particular squid is a nocturnal feeder. So, when this squid is like moving around in the sea water and looking for its food, probably there is a chance that it itself would be a food for its own predators. How the predators are going to locate this squid is through the shadow of this particular squid. And it is these bacteria, the Allovibrio fishery, which are actually going to provide the counter illuminating camouflage which I told. This squid, it is having a special light organ. So these particular bacteria, they are going to occupy this light organ there. Now they may also have, some squids, they have special reflectings also, some special proteins which will adjust to see that the shill outs of this particular squid are removed. So now the squid is moving around freely and is not casting the shadow, and that is protected from its predators. So it is illuminating, like it is glowing there, the bacterium is glowing there, and it is providing what you call as protection to this particular squid. Obviously it is not going to do this for free. No one does anything for free. So these bacteria also are not going to provide protection to these particular squids for free. What will they charge? They'll charge some food. So the squid is providing them with sugars, it is providing them with the amino acids. So like these bacteria can sit quietly there, they'll get a good shelter there, and they are getting good source there. So they are eating the sugars, they are eating the amino acids, they are growing and multiplying, and they are bioluminescing there. And the phenomenon of emitting light we call as bioluminescence. So here you will find matlab, the squid, when it is spawned, the bobtail what you call as squids, the hatchlings of these squids, they do not contain these bacteria actually. Matlab, so they are free from that. They have that special light organ, no doubt. Matlab, as soon as they are formed, they are born, they have these light organs. But these particular light organs are devoid of these particular bacteria. So they have to obtain these bacteria from the sea water. How it does is it is secreting a mucus whenever the squid, it comes in contact with peptidoglycan, like the substance which is present in the cell wall of the bacteria. The squid starts secreting a special mucus, and this particular mucus, it goes and accumulates at the mouth of the light organ. So all the passing by bacteria, they go and get trapped in this particular mucus. Matlab, you will be like surprised to know is that squid has elegant mechanism to eliminate all the other counter bacteria, but to keep only Allovibrio fishery. In many ways it does that. Like what it does is like the other bacteria actually cannot survive in mucus for a longer period of time. So automatically the other bacteria, they are eliminated. The light organ, it has special ciliated cells which are showing continued movement. This Allovibrio fishery also has a flagella and is motile, so it also provides a counter movement. So the movement of cilia actually cannot get rid of this Allovibrio fishery. So Allovibrio fishery can successfully colonize there, it remains there. So though the cilia are like beating and they are removing all the other bacteria from there, this particular bacterium, it still remains there. So like this is how it tries to see that it doesn't get any other what you call as bacteria in there. It is also observed that if Allovibrio fishery species are also there, but they do not show the phenomenon of bioluminescence, matlab, they also cannot survive there for a much period of time. So they do not bring about the changes in the light organ which are brought about by the bioluminescent counterparts. So this indicates that to colonize that light organ, that particular bacterium should compulsorily possess the phenomenon of bioluminescence. It is also found that the squid, it produces the enzyme which can degrade hydrogen peroxide. So it uses that as a weapon. So the hydrogen peroxide is degraded and all other bacteria which are there, they are killed because of that. Because of the toxic species which are produced after the what you call as breaking of this hydrogen peroxide. Whereas these Allovibrio fishery, what they can do is that they will capture this hydrogen peroxide before the squid can actually catalyze the degradation of that. So that also actually favors this Allovibrio fishery to stay there. So it's not easy actually for Allovibrio fishery also to colonize there. You will find that if you go and find out a squid, a Hawaiian bobtail squid and its light organ, you will find that only Allovibrio fishery is colonizing there. So after passing all these hurdles, like after surviving in the mucus, after passing out the ciliary movements of that particular cilia, and after say even counteracting that hydrogen peroxide kind of a phenomenon, these what you call as bacteria are present there and they are illuminating there at night and they are shedding light, and once they shed light they are removing the shadow of that particular squid and it's protecting the squid. Surprisingly, probably, you all will be surprised to know that the squid actually, sometimes the cephaloid squid, the other family of squid which has these bacteria, they remove around 90 to 95% of the bacteria from the body every morning. Sometimes it is every morning or sometimes when they are not needed. Now, this process of shedding these bacteria is called as venting. Now, sometimes we feel that why it is doing this venting, because at the first place it was very difficult for these bacteria to acquire them, to acquire that particular light organ, and now like it is shedding them. Probably venting, what it might be doing, it is hypothesized by the ecologists, like because of the venting probably the new hatchlings which are there, they should get the good supply of these Allovibrio fishery so that they are also protected. So they ensure that the upcoming generations also are colonized with this Allovibrio fishery. And probably another reason would be it's very costly for the squid to host these particular bacteria with it, because it is taking a lot of what you call as sugars and amino acid, and the squid has to keep on synthesizing them for these particular bacteria. So in the morning probably they are not needed, so why waste a lot of energy in sending there. So a wonderful, you can see, a mechanism, a very intricate relationship like of mutualistic symbiotic association can be observed in these Hawaiian bobtail squid and your Allovibrio fishery. I hope that you really loved this and enjoyed this information. It was new to all of you. It's not new actually. People are like molecular biologists, the people who are working with quorum sensing, they are using these what you call as mutualistic association as the model organism or the framework to study how exactly the bacteria are communicating with each other and with the higher animals. So yes, stay tuned with me, Professor Girish Kukreja, for more in microbiology. Thank you.